Hi, Professor Granlund here. I have some introductory comments about the cardiovascular system and the blood vessels more specifically. The cardiovascular system consists of the blood vessels, and the heart. In the broadest terms, the function of this system is to deliver nutrients, including oxygen, to the cells of the body, and to take waste products, including carbon dioxide, away from those cells. In other videos, the more specific functions of the blood and the heart will be described. In this video, we're going to focus on the blood vessels. Let's start with some definitions. Arteries are blood vessels that carry blood away from the heart, and veins are vessels that carry blood to the heart. Because arteries are usually shown in red and veins are usually shown in blue in diagrams, sometimes people think that arteries carry oxygenated blood and veins carry deoxygenated blood. And while that's usually true, it's not always true. For example, the vessels that move blood from the right ventricle of the heart to the lungs are arteries. They travel away from the heart. But those pulmonary arteries are carrying deoxygenated blood. There are anatomical differences between the arteries and veins. For example, even though they both have 3 layers called tunicas in their walls, the arteries have a much thicker middle layer of smooth muscle, and the veins have an inner layer that forms valves. As arteries move away from the heart, they branch like a tree into smaller blood vessels until they reach the vessels that deliver blood into the smallest vessels, the capillaries. Those vessels that lead into the capillaries are called arterioles. Small venules lead out of the capillary beds and collect into larger and larger veins. The capillary beds are where all the action is. It's the actual site for the delivery of nutrients and the picking up of waste. It's sort of like UPS driver leaving packages at your house at the same time that the waste removal truck is picking up your trash. Most of the capillaries in the body have small clefts between the cells to allow the exchange of nutrients and waste. And in some organs, those capillaries are designed a little different, like the fenestrated capillaries in the kidneys that allow filtration of the blood, or the sinusoidal capillaries in the bone marrow that allow blood cells to be exchanged. In the typical capillary bed, fluids and nutrients leave the arteriole end of the capillaries through bulk flow and diffusion, while fluids and wastes are returned into the capillaries at the venule end. The forces that drive exchange are the hydrostatic pressure, that is the capillary blood pressure. It tends to push fluids out, and the opposing osmotic pressure, drawing fluids in. The hydrostatic pressure is higher at the arteriole end of the capillary than at the venule end, and that's because there's still pressure in that arteriole end from the left ventricle contraction that forced the blood into the arteries. The osmotic pressure is caused by non-diffusible molecules such as albumin. So, with a higher hydrostatic pressure pushing fluids out of the capillary than osmotic pressure drawing fluids into the capillary, fluids leave at that arterial end. But as the fluids leave, the hydrostatic pressure drops. So at the venule end of the capillary, the pressure gradient favors osmotic pressure. The pressure is so low in the veins returning blood to the heart that it actually requires the adjacent skeletal muscle contractions, the respiratory pressure changes, and valves to get that blood, now deoxygenated, back to the heart. Let's take a closer look at blood pressure. Pressure is defined as a force per unit area. Blood pressure is the force exerted on the vessel walls. This pressure is highest in the aorta and lowest in the vena cava. When the left ventricle is contracting, blood is forced into the aorta. This is called your systolic pressure. It's typically around 120 millimeters of mercury. When the left ventricle is relaxed, blood's not being actively forced into the aorta and pressure is lower. That's your diastolic pressure, typically around 70 millimeters of mercury. Blood pressure is continually monitored by the body, and adjustments are made frequently. For example, when you stand up or lay down, when you're stressed or depressed or angry, or when you're exercising or you're ill, when you're smoking or drinking. The list goes on. The body can change blood pressure by changing cardiac output, peripheral resistance, or blood volume. This means that the targets for your regulatory system, the nervous system and endocrine system, are the heart, the blood vessels, and the kidneys. In another video, we will consider how blood volume can be adjusted by adjusting urine output. In a separate cardiovascular system video, we defined cardiac output as the amount of blood ejected from the heart per minute, and we mentioned that our regulatory systems can change cardiac output by changing stroke volume or heart rate. Here, we're looking at peripheral resistance. Peripheral resistance refers to the resistance to blood flow in the blood vessels, and one of the factors affecting it is the diameter of the vessel lumen. A large lumen means less resistance and less pressure, whereas a smaller lumen means more resistance and more pressure. Both the endocrine system and the nervous system can control that vessel diameter. Before we continue, let's recall that homeostasis requires a receptor, control center, and an effector. Very important blood pressure receptors, that is, baroreceptors, are located in the walls of the heart and the large blood vessels. And one of the major control centers for blood pressure is found in the medulla oblongata of the brain. Let's put a few of those things together. If the baroreceptors in the large vessels detected a decrease in blood pressure, they would send that message to the cardiovascular center in the medulla, and the medulla would respond by sending sympathetic innervation to increase heart rate, which would increase cardiac output, and to the arteries, particularly the arterioles, to vasoconstrict, which would increase peripheral resistance. These measures, increased cardiac output and increased peripheral resistance, would increase blood pressure. It is a classical example of a negative feedback loop. An increase in blood pressure would result in parasympathetic innervation and the opposite effects. It would decrease cardiac output and decrease peripheral resistance to decrease blood pressure. Several hormones could also be used. For example, if the baroreceptors in the large vessels detected a decrease in blood pressure and sent that message to the cardiovascular center of the medulla oblongata, the medulla oblongata can respond by sending innervation to the adrenal medulla, which would respond by secretion of epinephrine and norepinephrine. Epinephrine increases heart rate and therefore cardiac output, and norepinephrine causes vasoconstriction. These measures would restore the blood pressure. Other hormones, such as angiotensin II and thyroxine, also act to increase blood pressure. Not as many hormones are used to decrease blood pressure, but one example is atrial natriuretic peptide, or ANP. If an increase in blood pressure is detected by receptors in the atria walls, the atria secretes ANP, and the result is vasodilation and decreased blood pressure. This hormone will also act on the kidneys to increase urination and therefore decrease the total blood volume. At the end of the chapter, you'll be given an opportunity to name the major vessels and explore how they deliver nutrients and pick up waste throughout the body. You've already looked into pulmonary circulation between the heart and the lungs. You'll also look at general systemic circulation and specialized routes, such as the circle of Willis in the brain and the hepatic portal system associated with digestion. Well, that should get you started on those learning objectives. Please contact me with your questions.